I'm Alex, I'm a research fellow at the University of Exeter, and I'm basically interested in how plants, which are quite dynamic, interact with their environments to grow and develop. And from the title, you might realise I'm interested in endocytosis. So I think one of the first things we all have to remember, so I come from a mammalian background, so we're used to looking down on plants as a model system, is that actually they're incredibly dynamic organisms. So these are plants responding to light signals. So over the course of the day, they can follow the sun. So they have the ability to interact with their environment. But that's quite a slow response. They're actually able to respond to hormones in under 30 seconds. So in this movie, where you see the color purple, auxin has been applied to the root. And when you see white, it's washed away. So when there's no hormone, it grows. And upon the application, within 30 seconds, the whole root growth profile can change and dynamically alter in response to these stimuli. They also get eaten, and I love this movie. So you see a caterpillar chomping on it, and it's able to communicate to the rest of the plant, I'm getting eaten, we should probably do something about this. So just remember, plants are actually very dynamic, and they have to interact with various stimuli in order to grow and develop appropriately. And I think this is mediated by the process endocytosis. So this is my model system, Arabidopsis thaliana. And when it senses a stimuli, one cell senses this and communicates it to the rest of the plant. And this is mediated by receptors on the cell surface here. So once these receptors get activated by binding to one of these stimuli or ligands, it gets removed from the cell surface in this process of endocytosis. And basically, this process of endocytosis regulates the number of receptors on the cell surface, which mediates how sensitive cells are to various stimuli. Because if you have too many receptors, you can over-respond. If you don't have enough receptors, you under-respond. So it's critical for mediating cellular sensitivity. And actually, if we understand this process, we can continue to grow food and feed everyone on the planet, which is quite good. I've just told you how important endocytosis is, so you must be thinking, wow, we must know everything about this. But actually in plants, we really don't know much about this process at all. When we look in the mammalian system over 10 years ago, we know a huge number of the proteins involved. We know exactly what they're doing, when they're coming, and what their function is. And it's true of yeast as well. We also know a lot about the process. And when I started in plants 10 years ago now, it just makes me feel old, you can see our model isn't very complete. We don't know the number of the proteins involved. And even when we do, we have big question marks as to what these proteins are doing. And the reason for this is in mammalian systems in yeast, high resolution and quantitative imaging approaches are very common because they're quite easy samples to image. In plants, we have a big fat cell wall, so people don't like doing advanced and high resolution imaging because you get a lot of light scattering. So instead of doing this high resolution imaging approaching, a lot of this model is based on static interaction assays, which isn't great for studying a dynamic and transient process, and also the fact that these guys are homologs to these other model systems. So this is based on a lot of assumptions with very little direct evidence. So I wanted to understand how this process is working, and as I came from the mammalian system, I was interested in actually, is it similar to mammalians or is it similar to yeast? Because they have different biophysical properties and you have different proteins. So if we understand more about plant endocytosis, we can understand more about eukaryotic processes that are fundamental to all life. So to do this, we turn to fluorescent microscopy, and if you're in the cell biology practical today, you should know this. So we take a protein from jellyfish, it's called green fluorescent protein, GFP, and basically if we stimulate it with a blue laser of 488, we can detect emission in the green channel of 525. So what this means is we can take any protein of interest and stick a big light bulb on it. And using fluorescent microscopy, we can then look in a cell and see actually what this protein is doing in the live situation. So traditionally what people would do is epifluorescence. You just shine a giant laser into the sample. But what this means is that you stimulate all the fluorophores on all the organelles, and you end up with a big blurry image, which really doesn't tell you much, except that you have signal and it would be better to do a different type of microscopy. So we use total internal reflection fluorescent microscopy. So instead of shining the laser directly through the sample, we shine it at an angle to generate this total reflection. And the byproduct of this is you get this weak evanescent wave which penetrates into the Z direction only about 100 nanometers. So what that actually means is instead of illuminating all the fluorophores in the sample, you're only illuminating those very close to the cell surface and on the cover slip. So it's perfect for processes like endocytosis. And for a long time, people would say you can't do this in plants, but here is a turf movie. Instead of a blurry image of all the fluorophores stimulated, you're actually just looking at individual events of endocytosis occurring in the live, intact plant system. So now we had this cool way of imaging endocytosis at the single event resolution. It was kind of important to start analysing these. And the way people would do this traditionally was manually, which is an incredibly painful process, and it induces a lot of bias into the system. So we turned to MATLAB, and basically we use this unbiased detection algorithm based on the point spread function of the microscope. What that means is I don't tell the system what these spots are, it automatically and statistically tests them and tells me if it's a spot or not. Then we can link these guys together and create track histories. It's not the most exciting tracking because they're quite static, but we can pull out individual events and see what these proteins are doing on the cell surface. And because this is automated, we can get high accurate numbers of their lifetimes, densities on the cell surface, which tell us about recycling defects, and most importantly, the average behavior of this protein on the cell surface. So this is clatharin, and in endocytosis, you start with low levels of clatharin. So if you have a fluorophore on clatharin, you have low signal. And as this vesicle and coat forms, you get more and more of this protein accumulation, and you get this increase of fluorescence. And then when the vesicle leaves the field of illumination on the cell surface, you get the sharp decrease, which tells us it's probably involved in endocytosis. But that's great. You can tell a lot about the dynamics of the process, but I'm more interested in the mechanisms driving it. So to do this, we wanted to look at the proteins that might be driving endocytosis. And we developed this departure assay. What this is in essence is we image clatharin, just as I showed you on the previous slide, but at the same time, we image a second channel with a different fluorophore, like, say, this blue protein or this red protein. And then we align every individual event to this moment of departure, which is when endocytosis is finished. And this allows us to say which stage of endocytosis these proteins might be involved in. And this allows us to make better predictions about what these proteins are doing. So just to give you an example of what this looks like, this is a protein called T-plate. I'll talk about a little bit later. But in this green line, you can see it's arriving before the clatharin, so we know it's an important protein in the early stages of endocytosis, and it persists throughout endocytosis. And critically, we can do this for all of those proteins in our working model. Based on the biochemical interactions, people said a lot of these proteins were involved in the same processes, but if we look at their temporal profile, this guy has a very different profile to this guy here, which is only present at the start, and this one is present at the end. So even though they were found biochemically to interact, if we actually look at their temporal dynamics, they are involved in the same process, but at very different stages, which means they have different physiological roles. So with this kind of high-resolution imaging, we were able to make predictions about what these proteins are doing. I can't tell you the full story because I only have 15 minutes, but we made this prediction that this T-plate protein might be involved in membrane bending, because it's at the start of endocytosis. There's a lot more detail, but we wanted to test this accurately. So during endocytosis, the membrane has to bend to create this vesicle. We showed that it wasn't via actin, which was a surprise, but we needed a way to increase our resolution to actually look at the structure of endocytosis when we're disrupting the T-plate function. The way people normally do this in mammalian and yeast systems is by using TEM, which is an electron microscopy approach, where you can fix your sample and then cut it up into slices and use an electron microscope. But the problem is endocytosis is super transient, so if you fix the samples, for some reason these vesicles would fall off, so you couldn't image them. So we had to come up with a different way of doing it. So we developed this metal replication of unroofed cells. Basically what this means is that we take plant cells and we stick them to a cover slip. We unroof them, which is a very nice way of saying we rip them in half. And what that means is the only bit of the cell that's left is the plasma membrane and its associated structures like endocytosis. Then if we spray this with a metal like platinum, we can then take it to a scanning electron microscope. So instead of photons to look at our sample, we fire electrons at them and look at how they're scattering. And this allows us to get higher resolutions to look directly at the plant intracellular membrane. So this is a plant cell that's been unroofed and replicated with metal. And if we zoom into one of these plasma membrane regions, you can see that we get these honeycomb football structures. And these are the clatharin endocytic events. So we know that clatharin has this very stereotypical pattern when it forms that looks like these little footballs. And so at the time we were working on this, Daniel Van Dam was also working on T-plate, and he made this inducible loss of function T-plate mutant, the WDXM2 line. So what we could do is make these cells induce the loss of T-plate function, and then using this electron microscopy, we could actually see if we get these round structures or flat structures. So in our control conditions, you get these big, fat, round, happy vesicles. So you obviously have normal endocytosis occurring. And upon T-plate disruption, things aren't round anymore. You get these flat patches on the cell surface. So again, I've told you should quantify your imaging. So to do this, we turned to machine learning because we wanted to make 3D morphology predictions about these structures from 2D data sets. So the first thing we did is we used Neparian cell pose, which is a deep learning algorithm, where we trained the computer to automatically recognize these honeycomb clatharin structures. And then whenever we put in an image it had never seen before, it could detect these with like 98% accuracy. And that would give us the surface area of these structures on the membrane. Then to look at how curved these structures are, we take advantage of the fact that actually the gray value from SEM images is relative to protein density. So if you don't have any protein in your EM image, you have a low gray value because there's no protein density for the electrons to bounce off or stimulate. Then if you have a single layer of proteins where the clatharin has formed, but it only has a couple of proteins there, you get this slight increase of gray value. But when you have your deep endocytic structure, which is incredibly protein dense, you get this very high gray value. And if we plot this surface area against this curvature estimation in our control conditions, you get these SR structures, which we called small and round, which are our clatharin-coated endocytic structures. And when you have this T-plate disruption, everything is presenting as a flat kind of structure. So using these kind of techniques, we could then predict what these proteins are involved in, and then using the even higher resolution of the electron microscopy, we could actually then kind of confirm it. So again, one of the reasons we kind of had to develop these tools is because we wanted to study endocytosis. And to do that, we had to improve our resolution and our quantification and our robustness. So when we started working on endocytosis, everyone assumed that actin was involved in membrane bending, because in yeast, which have high turgor pressure, which is the opposing force of this bending, you have to have actin involved. So for years, people predicted that actin was involved in plant bending because you have even higher levels of turgor pressure. But by looking directly at single events of endocytosis at the high resolution live single event scale and at the ultrastructure layer, we showed that actually you don't need actin to do this. So it's actually evolutionarily distinct from all the other eukaryotic model systems we've looked at. And the other thing we've been able to do with these high resolutions is that we've redefined the role of the T-plate complex, which is a complex that's unique to plants. And it was predicted to be inside these endocytic structures, but we showed they were outside and actually involved this membrane bending. We've been able to look directly at receptors, how they're ubiquitinated and selected for cargo, which directly regulates how they're getting or mediating physiological pathways. And we've defined the proteome of these endocytic structures. So in the last slide, what I'm doing now is I'm continuing to work on endocytosis using these high resolution techniques. And I'm particularly interested in how different combinations and isoforms of these standard endocytic proteins can mediate it specific physiological events. And one of the other things I'm interested in is actually how the cell wall biomechanical properties, because this is affected by environmental stresses, can actually regulate the plasma membrane processes like endocytosis. And to do this, in my last postdoc in Vienna, we were building these Brilliant light scattering spectroscopy systems where we can use optics to visualize the live biomechanical properties of the cell wall when we're stimulating it. And we've just won an alert grant for 1.8 million to build this system in Exeter, where we can correlate this with fluorescent microscopy. So in summary, endocytosis is super important for plant physiology, but we don't know much about it. We've had to develop these tools to kind of investigate these questions, and in doing so, we found that it's evolutionary and mechanistically distinct from our model systems, so it is actually quite interesting, and we're beginning to understand how this is regulating different physiological pathways. So the acknowledgement slides, Christian taught me how to image, Gregory Wehr was the first plant supervisor I had, and Jerzy Frimmel in Austria we did a lot of imaging with and has supported a lot of my career. The MedUni and Exeter, we have our spectroscopy experts, and I just want to show you this. This is a 3D image of an endocytic structure in a plant cell, and if you have any questions, please ask.